All right, I'll go ahead to start. Good morning, everyone, or good day, depending on where you're joining us from. My name is Aigbidu Olufunke, and on behalf of the APSS Organizing Committee, it is my pleasure to welcome you to our fourth webinar. And before we get into the webinar proper, please go ahead and drop a message in the chat box telling us where you're joining from. Would love to hear from you. Just a couple of reminders for those that just came across it. We're talking about customizing your field book without any programming knowledge, and we have two amazing speakers. Before I introduce the speakers, a couple of reminders again is that our symposium is coming up on the 18th, on the 19th of September, and abstract submission deadline is coming up really soon. That's about 9 days from now. August 31st is the deadline for submitting your abstract. So if you haven't submitted your abstracts, please go ahead and do that now. We have both graduate and undergraduate competition, so you would love to be a part of that. And with that, I would go ahead to introduce our speakers. Dr. Leo Gasper, he grew up in a farming family in the Philippines and he earned his bachelor's degree from the University of he earned his bachelor's degree from the University of Philippine Los Baños in agronomy and plant breeding, and he began his career with the Philippine Rice Research Institute and later in the International Rice Research Institute, working on rice breeding, golden rice, and nutrient-enriched varieties. He then spent about a decade at DuPont Pioneer, leading hybrid rice trials and breeding programs. Dr. Leo Gasper went to pursue his PhD at Louisiana State University and continued his research at the University of Florida, focusing on trials enabling mechanical harvesting in fresh market tomatoes. He is now a plant breeder at Lipman Family Farms, where he applies predictive breeding to develop high yielding, disease resistant, and high quality tomato hybrids for production across the southeastern United States. And our other speaker, Dr. Sherry Dalid, is an assistant plant breeder with the University of Florida Strawberry Breeding Program. She earned a PhD in plant breeding with a minor in statistics from the University of Tennessee and a master's in genetics from the University of Philippine Los Baños, where she also worked at the International Rice Research Institute. At University of Florida, Dr. Dalit develops strawberry varieties for Florida, Southern California, the Southeast United States, and international markets. She has co-invented four University of Florida strawberry varieties and three white strawberry varieties, and also leads the Strawberry Phenomics Project, pioneering digital phenotyping tools that advance growth modeling and biomass measurement in strawberry breeding. So thank you very much for agreeing to join us on this webinar. And with that, I will hand it over to Dr. Cheryl Dalit for the webinar proper. Thank you.
Okay, thank you for the introduction. Let me share my screen first. Okay. Good morning and thank you for that introduction and thank you for inviting me. So this morning I'm going to present the digitizing data collection in the UF Strawberry Breeding Program. So before I start, I just want to give a brief background or brief introduction about strawberry production here in the United States. So majority of the production of strawberries comes from California, and this makes up around 59% of the total production in the US. The second largest producer in the United States is Florida, which makes up around 11%. And the rest would be other states like Oregon and Washington State. So this next slide, it just gives you an overview of all the activities that we do here in the strawberry breeding program. So we start our planting for fruit production from September to October. And after the plants establish and they start producing fruits, we do all our data collection from November to March. And this is the time when we also do all our field selections and all our crossing activities in the greenhouses. And by April to May, that's when we start our marker assisted seedling selection, where we grow all the seedlings that we develop from our crosses, and we screen them for molecular markers. And whatever we select, we plant in our summer nursery in Oregon for plant production, uh, in preparation for another season of the fruiting production here in Florida. So why did we start using apps for our breeding program? So when I started here, they already have a system. They have tablets. And with these tablets, each individual person taking the data can take a tablet. It has the Excel file. But the problem there is, as someone who has to consolidate all these different Excel files, different weeks of harvest. It's really time consuming and it's definitely a lot of work. So we have to find a way to make it more efficient for us. And that's where um, we found Phino Apps, which is a suite for open source plant breeding apps. And within the Pheno apps, there are several different apps that plant breeders can use. But the most commonly used, I believe, is the Fieldbook app, the app that you can use for your data collection. And for our program, we also use the Intercross app. And so, what's the importance of these apps that we are using? So, we need data integrity, right? With all these new technologies that we have, like hyperspectral imaging, multispectral, we handle a lot of data, and we have to make sure that we have data integrity. Our data is accurate, our data is consistent. Second is data security, because as I mentioned, we have different tablets before and with different Excel files within those tablets. And those are easily lost because sometimes someone forgets to save the data, or we cannot find which tablet we use for a certain day. So there's a lot of things that happens between data collection and getting all those data consolidated. And we also need to have a good data turnaround time. That's with the app, you can just take the data, and within the day, you are able to see the data. You can even do some preliminary data analysis or even just check your data for outliers. And again, for this is also important for data curation. As I mentioned, consolidating all these different Excel files from different persons, from different weeks is time consuming. And so having just one app that can consolidate everything into one database is definitely helpful for breeding programs, especially ours also. So with the Fieldbook app, there's a lot of data type that you can collect. You have the numeric data, categorical. You can even have your dates or disease ratings, and you can even take photos of uh, your materials. And this is something that we've used in our field data collection as well. And another app that I mentioned is the Intercross app within the Fino app suite. And this is also something that is very useful because it gives you a summary of the number of crosses that you've done in one day. So we started by introducing the barcode system, where barcoding or the QR codes, where we use it to make sure that we have all the genotypes that we need in our greenhouses. In addition to that, as I mentioned, we've used the Intercross app for our crossing. And before, we have a system already where we use Microsoft Access to have all the crossing uh, information. And we actually just have like one laptop that we bring to our greenhouses, and it sits in the middle of the greenhouse. And whenever we have to enter how many emasculations we did, how many pollinations, we have to go back and forth to that um, laptop, which is also time consuming. And so we had to use the Intercross app so that we can just have individual tablets that we can use, or phones, and it works perfectly. However, our program has different needs as well. And the problem is we have to keep track of how many flowers we have to emasculate and pollinate, not just the different combinations of crosses. But at least we already know that it worked, and we also use the barcode system in our crossing greenhouses, because this barcode serves as our unique identifiers, something that is very important when you're using Fieldbook and also the Intercross app. And as I mentioned, our program has certain needs that you also need to make sure that we are addressing. And so we have to find another way to improve our system. And just wanted to mention that there's a lot of different platforms that you can use for phenotyping. But you also have Power Apps. And in our case, we used AppSheet, which is a non you don't need to know coding to develop the app. And for this, we used it in our crosses, and this helps us with keeping track of how many emasculations, how many flowers we emasculated, and how many pollinations we did in one day. And because it worked well with our crossing greenhouses, we even moved to our selection field, where when we go to the field, we just take our phones, and whatever we select, we can see the pedigree, and we can even add notes to our selections. And eventually, we use the same platform for all our data collection, uh, all our weekly data collection, from our yield trials to our disease trials. And so far in our breeding program, we've fully integrated all these apps, where all the activities that needs to have data, we've used or we've developed these custom made apps, which makes our life a lot easier. So just to give you an idea, these are the gadgets that we've used. Something that uh, will be useful if you use the field book and the Intercross app, and at the same time, it also works with the custom made apps that uh, we've developed. And with that, I'd like to just acknowledge the whole team, which has been very supportive in all these endeavors that I've done with the program of using these apps for our data collection. And with that, I would like to thank everyone for listening, and thank you so much for the organizing committee for inviting me.
Thank you very much, Dr. Dalit. I think we are having Dr. Gasper next.
Yeah, yeah. Let me start my presentation.
Okay, and if you have any questions, please drop them in the comments in the chat box, and we'll attend to them at the end of the presentation.
Yeah. Okay. So, Doctor, you all can hear me well, or okay. So what Dr. Cheryl Daly shared to you all is experience in the academe, in the public sector, right? So my career has taken me, most of my career has been in the industry. So I'm gonna share a little bit of that experience. And towards after 10, 15 minutes, we're gonna go to an exercise that will let everyone have the experience of setting up their field books in their own devices. So again, good morning, or good evening, good afternoon. I saw that there are a lot of participants from around the world. So I hope you are doing well today. Again, I'm Leo Gaspar, and I'm presenting today not only on how to use the Fieldbook app, but about my experiences about research data collection in my years of working both in the industry and in the public and or academia setting. And just to be clear, I'm not a subject matter expert about the Fieldbook app, but I am presenting as a longtime user of electronic data capture apps, which including but not limited to the Fieldbook app. And I don't intend this seminar or workshop to be very detailed, but what I'm hoping to accomplish is to kind of whet your appetite about the capabilities of what this app and what it can help you or how it can help you organize the way you record your data. And this is not only for plant breeders. I'm pretty sure there are plant pathologists, entomologists as well, or soil scientists in the call. So this is not limited to use for plant breeding experiments. And during my 10-year stint at Pioneer Hybrid or DuPont Pioneer, the company provided both software and hardware for us to record our data electronically. So field notes and observations were recorded using their own native app. They call it IDC or iOS data collection. And they also provided printers and scanners and software that enabled us to scan barcodes and QR codes. So barcodes, QR codes, it's not really required for you to record your data electronically, but it makes your data collection way more efficiently. It avoids errors. So the next few slides, just going to show you how we implemented barcodes or QR codes. So we barcoded or QR coded our plant tags. So I work primarily in rice. So we barcoded our rice seedlings even in the seedbed. And that particular barcoded plant tag followed a particular genotype from the seedbed to the field and up to maturity, even after harvest, if we needed to do some post-harvest testing. And I say barcode or QR code, but you can see here, this is a data matrix. So you might hear me use those terms interchangeably. So just barcode as a general term for anything that you can scan, be it QR code or data matrix. So that 10 years at Pioneer, I was so pampered, I guess, and I got used to recording my data that way that it felt so wrong for me every time that I needed to record my data on a piece of paper. So the system was very efficient, it was seamless, but the caveat is that the software was proprietary. It was developed by the company. It is only available for people working at the company, and only those people has access to it. So fast forward to the time I started my graduate studies, graduate program, first at Louisiana State University, and then later on at the University of Florida. I don't have that system. I don't have that capability to record my data electronically. So just to share this, just to share the system that we had at LSU, the hybrid rice breeding lab at LSU. So everything was done manually. data collection, field data were all handwritten on a piece of printed Excel sheets. Seed inventory information were handwritten. In fact, I took this picture in our lab, and one of our technicians, like, this is kind of a protocol, like for us to write our seed inventory information. ID, we have ID line 1. Let me use my pointer. Okay. Let's we have here where we're supposed to write line 1, line 2, the date where we harvested it, when we harvested it. And then it also has a guide, like how do we write 1, how do we write number 2, 3, 4, 5, 6, 7, 8, 9, 0, for us to be able to kind of standardize how we write those numbers. It was kind of not really shocking, but it was a big change for me coming from everything is electronic, everything is printed, to going back to handwriting hundreds, if not thousands, of flat tags and seed inventory packets. And sure enough, it led to some confusions like this. So you have things like you have seed packets that one person will read it as 899 and the other person will read it as 668. So which is it? So things like this is very common when we write inventory information manually, or we write on a piece of paper because one person writes differently from another person. And luckily at the time, the inbred rice breeder, his name is Dr. Adam Famoso. He's still at LSU. At that time, he was in the process of implementing the use of Fieldbook app in their program. And I convinced my professor then, Dr. James Ord, to implement the same system in our lab. So this is how I came across the Fieldbook app, or the Fino apps in general. So Fino apps is like what Dr. Shell mentioned earlier, refers to a collection of open source, user-friendly mobile apps that can help initially just plant breeders, but anybody can use it. Any person that needs to record data can use it. And Fieldbook app was developed by Dr. Trevor Rife when he was a student at Kansas State University. Dr. Trevor Rife is now an assistant professor of phenomics and crop improvement at Clemson University. So he has this, they have this publication, and you can read all about the Fieldbook app. And it was published in 2014, but since then they have made tremendous improvements in the app. Yeah. And this is how just to show how we implemented the Fieldbook app and related resources provided by the same group, the Fino apps group. So instead of writing down plot tags, hundreds and hundreds, if not thousands of them, we printed our plot tags using whatever resources that we have at the university back then. So we didn't require a specific printer. We used the ordinary laser printer that we have at the office. We bought weatherproof sticker papers. So we printed our plat tags instead of writing them down, all the details by hand. So we also use barcode meter sticks to measure plant height and panicle length data, or flag leaf length data. Barcode was also used to record flag leaf angle. So I work in hybrid rice, so flag leaf angle is important as far as hybrid seed production is concerned. So we use that. And there's also a separate app that was developed by FinoApps, and I think Dr. Cheryl Dallin mentioned that earlier. It's called the inventory app. So seed yield and inventory information, we started updating them using this digital scale. At that time, it cost like $25. I mean, that digital scale and the barcode scanner close to $50, I think. And the cell phone that we use doesn't cost me like a couple $100, I think. And all these changes that we implemented using Fieldbook app, using inventory app, this allowed us to minimize human errors in data encoding, and it resulted to faster data turnaround. If you are a graduate student, or even if you're not a graduate student, if you are managing a research program, you want your data analyzed real fast, right? So if you have your data handwritten in a piece of paper somewhere, then you still need, or you still require some time to encode the data. When using Fino apps, Fieldbook app, then your data is ready for analysis as soon as you are done collecting your data. So this system, the implementation of Fino apps, Fieldbook app more specifically, it allowed us to analyze our data faster than our previous system of handwritten notes. And like I mentioned, we upgraded our way to manage our data, our seed inventory. So we started printing QR coded inventory information instead of writing them down. All our field harvests were now labeled with QR coded tags, not by handwritten plat tags. So now that you have a glimpse of what can be done, the question now is how to implement the use of the Fieldbook app. And I must mention that unfortunately, or yeah, that Fieldbook is only available for the Android devices. But although the developers mentioned that an iOS version is already in the works, but they mentioned that it might not be available yet in the next couple of years. So hopefully they'll come. They will publish an iOS version of the Fieldbook app soon, because it's a really good app for data collection. So I summarized the major steps and how to implement or how to use the Fieldbook app. And basically, these 5 major steps, you can be more granular or more detailed about each step. But these major steps, once you accomplish them, it will get you ready to record your own research data using the app. Okay. And I'm going to briefly discuss each of those steps, and then we will have a hands-on exercise later, so each and everyone can have their chance of practicing using their own Android device. Okay, the first step, you're creating a new data sheet for Fieldbook. So usually it's a USB file, or sorry, a CSV file or Excel file. So there are 3 unique columns, and this was in the previous version of the Fieldbook app. Right now, I'm seeing maybe they only require the plant ID. So plant ID is something that should be globally unique across all your experiments. So it could be your plot number. It could be, say, for a plot in Los Baños, Philippines during the rainy season, it could be WS for wet season, and 25 meaning 2025, and then a location, Los Baños, LB, and then whatever field number it is, like 41. And that's something that is unique. It won't happen again because you'll only have an experiment in only, like, repeat that experiment only one time in Los Baños during the wet season of 2025. Next year, it will be dry season 2026 in Los Baños, Philippines. So it will be a different ID. Okay, the next 2 columns, this is just to guide you navigating in the field as you collect your data. So sometimes they call this range and plot, sometimes they call this column or row, sometimes they call it X and Y coordinates. So the way this is how you move in your field. So if you move from like within a column, but you move between between rows. Okay. So how whatever system that you use to arrange your plots in the field should be reflected in this column here. And the other columns are optional but needed, right? Like what's the treatment name? It could be the treatment name could be a variety. In this case, these are different rice varieties in LSU. So you could put pedigree information, you could put anything that you want. Treatment. Okay, so all these are optional, but as researchers, we need them to identify treatment from one another. Okay, so that's the first step: creating a new data or a new field for Fieldbook, and that is done in Excel. So you save that as either XLS or CSV, and you download that or you upload that sheet into your device, into your phone, into your tablet, as long as it is Android and you have the Fieldbook app installed, then you can do that. Okay. So when you connect your tablet into your uh, laptop computer or your phone into your computer, this is the file structure within that Fieldbook app. So there are 8 folders, but I'd like to draw your attention to these 2 folders first, the field import and the field export. Field import is where you put your newly created fields. So that Excel sheet that you created in the previous step, this is where you put it, field import. And once you are done collecting your data, and then you export your data, then this is where you get field export. Okay. And one other folder that I'd like to discuss is this resources right here. So a lot of us are, especially when we are starting a career or starting a new career and starting with a new project, new experiment, we need visual aids for us to be able to standardize our scoring. So usually it's like a standard evaluation system for rice, standard evaluation system for tomatoes, things like that, and they're usually in PDF form or pictures. And you want to access those references while you are out there in the field, then you can store those PDF files or pictures in this folder here so that while using the app, you can open them in the field. So I think that's one of that helped me a lot when I was using the app. Okay. So after uploading, by the way, you can do your uploading by USB. You connect your tablet to your laptop. But for me, the most convenient way is through cloud. So if you connect your if you connect your cloud account into your tablet, then when you open your Fieldbook app, you can download data directly from your cloud account, Google Drive or whatever cloud services that you use. Okay, I think this is Dr. Dalit showed this briefly earlier. So you can take various types of data using Fieldbook apps. It could be numeric example, numeric data. One example is height. In rice, we also count our tiller numbers, so we can use that as when we create a new trade. You can use categorical type of data. It could be stem color for us. In rice, it's either purple or green. You can also use a data type whenever you are recording a heading date, emergence date, things of that nature. So all the things that all the data that we collect as plant scientists, plant breeders, plant pathologists, entomologists, soil scientist, um, it's in there. If you don't find the right data type, you can always send a note to the developers and make a feature request. So once you install the app in your tablet, it has kind of a default set of traits, but you can create your own trait, ad hoc traits. Like as you can create traits as you need them. Yeah. So this a couple of slides ago, I mentioned resources that you can put inside the app so that when you go out into the field, you can say, "Hey, what is a 1% uh, like for a streak disease, for example, bacterial leaf streak? What is 5%? What is 25%? What is 50%?" So something that you can use in the field to help you standardize your scoring system. So this is not specific to rice. You can do that for cassava. You can do that for sweet potato, corn, what have you. If you have that kind of an illustrated guide, it could be identifying diseases, right? So you can put that inside your app, and it can definitely help you standardize your scoring or identify diseases in the field. So one feature that I like about a Fieldbook app is the user profile. You can set the name of who's the researcher collecting the data. So for me, that promotes ownership. So I'm proud that I am the person collecting the data that is attached to my name. I better be making good on my scoring, right? So it promotes accountability, it promotes traceability. Like you can go back and, hey, we have so many outliers. Is this really what we saw in the field, or is it because of a particular story about? So is it because of actual differences that we saw in the field, or is it because of a particular person that probably needs more training, right? Give me 1 minute. Okay. Okay. So this is what it will actually look like when you are out there in the field collecting your data. This is when you actually collect your data. So the recent update that they made updated this user interface. It still looks almost the same, but they added a lot more features. So when you open your newly installed Fieldbook app, don't be surprised that it looks a little bit more different, but it's basically functioning the same thing. So you can have your menu to go back to your Fieldbook, to your traits, things like that. You can also click this button here for tutorials. You can scan your barcoded plot tags to jump to a particular plot number. You can search for a particular a treatment name, and this is where you can access those resources or guides, illustrated guides that we were discussing earlier. And you can click this icon here to look up all the information related with this particular entry right here. So the way I set up this particular field book is my unique ID is my plot ID, so 2019 S1-1, but that particular plot is associated with this particular um, uh, genotype 16SF240-1. Okay. And then during this particular run, I'm um, measuring height, and I'm actually in column one, row one. You could name it whatever convention that you're using. So it could be X and Y, it could be range and plot, things of that nature. And when you are collecting your data, you can stay in that same plot and collect all the data that you need. So it could be height, it could be Taylor number, it could be anything, disease rating. But for my own preference, I usually like to um, get my data for a particular trait and then move to the next plot. So you can move from trait to trait within a plot, or you can move from plot to plot and measure the exact same trait. Okay. And then once you are done collecting your data, you can now go ahead and export your data. And it says here you can export it as a database and you can export it as a table. My preference is just export it as a table. So main difference between database and table is if you have any experience in managing your data set, you know that there is a long way of formatting your data, more ready for analysis, R and other software. So long form is the database, and the wide form is your table. For me, I always like to export my data as table. Sorry. And you can export all your imported columns, meaning all your data, and you can also export only your active traits, meaning all those columns or traits you have data, or you can export all your traits. And then the way it creates a file name is it has a timestamp, so you avoid losing information also. So you'll have that timestamp associated with your data export. Again, you can export your data by physically connecting your tablet into your laptop or into your system via USB. You can send an email with the data as an attachment, but my favorite way of saving my data is via cloud. So I have my Google Drive associated with my Fieldbook app. So I can just, whenever I have a Wi-Fi connection, I can just um, download into my cloud account. So I don't have to bother with USB cable and stuff. Okay. So now we move on to our hands-on exercise, and hopefully you'll have your Android devices with you. We can do this together, and we will. The four steps is to create or modify and upload a field. If you already have the Excel file that the organizer sent to you, we can use that, or we can create from scratch. So the way I'm gonna do it is I'm gonna create one from scratch. So we can do that together. So after creating and uploading a field, we can define the traits of whatever plant height data or panicle length data we want, record a sample data, and export that data. Okay. So let me show you my sample data. And I hope you're seeing my Excel sheet now. Are you seeing it?
Yes, we can.
Okay. So this is just an example of a data sheet that I can use. Say, for example, I like my unique identifier as my EU ID, meaning experimental unit ID. And I know I have 8 entries, so I'm just gonna drag down. So for me, it's wet season of 2025 in Los Baños, Laguna. and Laguna, Philippines, and then I'm starting with 5001 plot number. So 5001, 5002, 3, 4, 5, 6, 7, 8. And the way I like to arrange my plot is I have a range. It could be your column or X coordinate. So I have two ranges, and within a range, I have four plots. So I now have range 1 and 2, and within a range, plots 1, 2, 3, 4. And let me type in, since I work in the plant breeding, in plant breeding, rice breeding, let me type in particular or popular rice varieties in the Philippines. Let's say Mestizo 1, 2, 3. And this uh, treatment names here could be a fertilizer, could be anything. This is screens or whatever you're doing in your own research. But for me, it's all rice varieties. And you can add information, other informations for plant breeders. They usually like to put pedigree information. Let's say 580025A by IR 34686R. And let me do this second row and move on. um, and you can populate the rest. So as simple as that, I can now save this as a CSV file. So right now it is saved as a CSV file. Save it. And you're done. You're ready. You are ready with your field. So I have that field book, that field in my computer. So the next step is to bring that particular, it's this file right here. So now I can either transfer it to my tablet via USB. But again, for me, the most convenient way is to just put it in my Google Drive. Okay. So I'll transfer it in my Google Drive, and now I can access that particular field in my laptop. Oh, sorry, in my field book. So everybody following so far? Okay. So now let me share my tablet, the screen of my tablet, and this is where all the fun begins. Let me just give me a minute to connect my tablet. Let me stop my share. For okay. Okay. You should be seeing now my tablet. This is the mirror image of my tablet. So within the Fieldbook app, you have your fields. This, I'm not sure if you can see my cursor now, but at the topmost, it will be your fields. Next is traits, collect, export, settings, etc. Okay. So the first thing you need to do when you want to upload a new field is to click the topmost icon, which is your field. So let's do that now. So a newly installed fieldbook will have sample fieldbook fields in there, but we don't want to use those, so we need to add a new one. So we click the plus icon at the bottom right. And for me, I'd like to upload that particular data or particular field from my cloud account. So I'll click cloud storage, the middle one, and then it will take me to my Google Drive, and it's the one on the rightmost file. So I'll click on that, and then it will ask me what is your unique identifier. I mentioned earlier I like to use my experimental unit ID as my unique identifier, so I'll choose that. You can choose different things, but again, for me, it's the EU ID. Okay. So say import. So now you can see at the topmost list, the field and the list, it's RPY, RP meaning Philippines, Y means yield trial, 25 means 2025, 2 means it's our wet season, it's our second season in the Philippines, and now I'm in my 12th field in Las Bañas, Philippines. So I can directly click that, but I still need to define the traits that I want. So you have your field book already. Let's go back. Let's click that back arrow to define the traits that you need. So for me, so the way to do that is to click that icon for traits. So let's go ahead and do that. Traits. Like I said, there are a number of sample traits already right there. But the trait that I want to collect is not in that list. So how do I create that? So I can click on the plus icon on the bottom of the screen, and then that will lead me to this window that says create new trait. So I can create new trait, and the trait that I want to collect for this particular exercise is flag leaf length. So it is a numerical data. So I'll choose numerical with the icon 1, 2, 3 in the middle. So I'll do that. And then I enter the name of the trait. So for me, flag leaf length, oops, FLL. And then you can put a default value that is optional. But you can also put minimum value. This is important because there are some values that are not really would be kind of outrageous or outlier. So this is a way to minimize your errors whenever you are out in the field collecting, or you ask someone to collect the data for you. You know that there are some minimum values, say for flag leaf length, let's say 10 centimeters is the smallest flag leaf I've ever seen. And the maximum is, say, 50 centimeters. So you can set the minimum and maximum. And you can put the details like what's your unit, how do you measure it, things like that. So you can save that, and you can add more traits as you need them. So right now, all these traits, they have a box with a check. So meaning when you open your field, now all this will be showing in that. So I don't want that. I just need, say, height, color, and then the new trait that I added, which is flag leaf length. So I'm gonna uncheck all those other traits that I don't need. You don't need to delete them. You just need to uncheck them. Okay, following so far. And for example, I want to measure my flag leaf length first. So I can also modify the order of the traits on how they appear in this list and how they will appear later when you are collecting your data. So you can just long press and then transfer it to the place where you want it. So I said I want to collect the flag leaf length data first, then I put them at the top of the list. So you're done with that, setting your trait, and now you can start collecting your data. So now you can go to collect data, the third icon on the list. You collect your data. Okay, sorry. So let's back up a little bit. So in the list of fields, you have 1, 2, 3, 4. Okay, 4 fields. So it could be location. In my example, could be locate. The first could be location in Las Bañas, Philippines. Next one is in Nueva Ecija also in the Philippines. The third one could be in General Santos in the Philippines. So you have all these fields, all these experiments. So you need to choose which one you are recording at that particular day. So I'm interested in recording my data for the Las Bañas experiment. So I'm gonna click that. And from there, you can collect your data. Okay. And within this screen, it has all the summary. It can tell you if you have exported your data. It can tell you if you've already collected the data. It can tell you a quick summary of your data. So let's proceed with data collection now. So you collect, and now it's showing you flag leaf length because that's the first trait that I wanted to collect, right? So flag leaf length, let's say for the first plant, it's 15 centimeters. So you collect that data, and then you press the black arrow to the right. That's how you move to your next plot. So you can see all those information up top. GE name changed from Mestizo 1 to Mestizo 2, and then your plot changed from plot 1 to plot 2. Okay? If you want to stay in the same plot and record all the data that you need to record in that particular plot, then you click the green arrow. So that's when you want to stay in the same plot but cycle through all the traits that you need to record. So you click green. So now I'm measuring plant height, and for plant height, I know that 100 centimeters is common. And move to the next trait. Color, let's say, for example, color of the stem. I know that particular hybrid is green, so I'm gonna click green. Okay, and now I'm ready to move to my next plot. Things like that. But I think I mentioned to you earlier what my personal preference is. I like to measure one trait at a time and move through all those plots and then move to the next trait. So once let's say, for example, I'm done collecting my data, or let's say you are curious, like, have I collected all the data? You can go back and see kind of some summary about your data. It says here for my flag leaf length, I only have one observation. For plant height, one observation, and for color, I only have one observation. And it gives you kind of that percentage right there of how much, or like an idea of your progress and how many more you need to collect. And going back to that collect data interface, if you go to the top, the icons with the grid, like boxes, if you click that, it will show you kind of a spreadsheet representation of your data. So here you can clearly see that I've collected all the data for plot WS25LB5001, but I have not collected any data from the rest of the plots. Okay. Again, that reference, I say, let's click that, the folder with the star. Let's say, I don't know how to score StemRes. And yeah, it now shows you my visual guide on how to score StemRes. You can put all the information or all the scoring guides that you need for your particular application. Okay, so now let's say, for example, you're done collecting your data. You can now go to your export. What it exports, by the way, what this field or what this icon will export is whatever the active field is. So when you go back to your fields, it will only export that RPY252 LB12. So go to export. Again, I mentioned earlier, I always like exporting my data as a table. You can export it as a database, like it's more amenable to straight analysis using R or whatever your favorite statistical analysis software is. I just like to export my data in a tabular form. So I can import all columns and then all the traits that I want. And then it creates, again, like I mentioned, it creates this timestamp when I downloaded the data, year, month, day, and even the time, minute, hour, minute, and second that I exported the data. And towards the end, it gives me the name of that particular field. So I can save it. So when you save it, it has that nice pop-up that reminds you to please cite the original article. That article written by Dr. Trevor Reif. And now you can see at the bottom, you have a different choice. You can export it using your as email attachment or as a you can save it in your Google Drive. So for me, I want to save it in my Google Drive. Okay. Export. Let's do that again. Okay. Okay. So now this is connected to my Google Drive and I can just upload the whole thing. Okay. So if I go back to my Google Drive, then I should have that data, that field with my updated data. So any questions at this point?
Dr. Gaspar, I think we can finish first and then we will ask the question. I'm
Okay.
collecting the question. Yeah.
Yeah. So that's the end of the hands-on exercise and the presentation itself. And hopefully you all learn how to do this on your own. If not, I'll provide a link to resources, kind of a more detailed tutorial on how to do this on your own. It's on YouTube, and they've done a tremendous job in explaining and how to do this in a more detailed fashion. Okay, let me stop my screen share so we can go ahead with the questions.
Okay, thank you, Dr. Dalid and Dr. Gaspar, for your respective presentation. I hope our participants have gathered a lot from it. And let me start with the first question. So please, how do you create thread list on your system before importing it to your system? That's the first question.
What was that? Sorry, I didn't get it. How to?
How do you create thread list on your system before importing
Oh,
it?
trait list. Yeah, yeah. So I can do that. I'm pretty sure you can do that. They say it has a TRT. It's basically the file extension is TRT, but it's basically a Excel file also. Personally, I don't do that, but there is a way to do that. If you don't know how to do that, you can go into your Fieldbook app and you can export your traits and then use that as a template for you to add in new traits. I hope that answers that.
Yeah. Okay, the second question, can these apps works globally or just in the US?
It works everywhere.
Okay. The third question, thank you, Dr. Cheryl, for that brief and detailed presentation. Your research work sparked some interest in me. Do you work on drought stress tolerance in strawberry? If you do, I would like to work with you with your team in this area of research.
Can you repeat that again? I didn't catch the
Your research work sparked some interest in me. Do you work on drought stress tolerance in strawberry?
tolerance.
Yeah, drought stress tolerance.
I think
Drought
that
stress tolerance. No, we don't. Just
Okay.
because in Florida our system is really, I'd say, strawberries are a high value crop. So we do use a lot of water here, and our soil is sandy. So we really use a lot of resources. But of course, for growing strawberries in a different part of the world, I think that is something that needs to be studied also. But at least in our program, we haven't touched that.
Okay. Thank you for your response. Then the next question, how do you import the CSV files from your phone storage to the Feedbook app? I think that one has been covered. Dr. Gaspar?
Yes. So it's basically the same process. So you go to that location icon or fields icon, and then you click the bottom right. There's a plus sign in there. So once you click that, you can go to instead of what I did was go to a cloud storage. So the other option is go to your local storage, and then in there, whatever is stored in that folder, in that import folder, you can upload into your Fieldbook app. So let me share again.
Yeah.
Which screen are you seeing now? Is it folders or no? Are you
Oh,
seeing a screen with folders?
yes.
Okay. So I connected my tablet to my laptop. So this is that file structure I was talking about. So this is your PC. Let's say Galaxy tab, internal storage. You see Fieldbook files, and then you can put your newly created fields or your CSV file, Excel file into the field import right here. And once you've done that, you can access all these files using your um, uh, Fieldbook app. So you can choose a local you can choose to open your field from a local storage instead of the cloud.
Okay, thank you. Then next, how to create barcode for multiple accession evaluation for crops
Okay.
that did not have a database such as okra, eggplant, and some leafy vegetables.
Okay. So barcode, so usually it's already outside. In my experience, I don't create, and I don't know how to create barcodes in the PhenoApps environment, but what I use, I can do Google Sheets. Let me check my my presentation. It could be, let's stop share and then share again to screen two. Okay. So this third bullet here, Avery Design and Print, you can go to their website and upload an Excel file containing your experimental unit ID, accession number, whatever, your unique identifier. And you can create QR codes, barcodes, data matrix, and they don't charge you anything. It's free. You can create those barcodes and save it as a PDF and then print it on your local system. I'm not promoting Avery because this is a company, but that's what I use. They have their own standalone program too, but they discontinued that already. But I still have that in my system, so I use that. There's another app called, let me check if I can open it for you. Um, it's called Go Label. So Go Label is a software that accompanies the uh, okay, it accompanies a printer that we use, so I have that for free, but they need to buy that particular printer. And again, I'm not really advertising that or advocating for that. So whatever system that they have, they can use that. And I think if they search Google, they can also create barcodes now using Excel, but I think it's easier to do that in Google Sheet.
Okay, thank you. So the last question from my end is the following. Thank you for a wonderful presentation. During the course of the presentation, I had a glitch with network. Can you please shed more light on the addition of traits?
Addition of traits.
Yeah.
Okay. Do you want like for me to just discuss it or you want me to show it in the tablet?
I think if you can show it will be good.
Tablet? Okay. I'll do that. Give me 1 minute to connect my tablet.
Okay.
Yeah. Yeah, I think it's better to show it in the tablet so everybody can follow. Okay, it should be coming up shortly. Okay. Okay. So give me a particular example of trait that you want to collect, for example. Let's say trait. You click trait and this all this in this menu, you click the icon that says trait, and then at the bottom right, there's a plus icon. So you can click that, you can click the new trait, create new trait, and then depending on your trait of interest, let's say stem color. I'll go, stem color is categorical data, right? For rice, could be purple, green. So I'll choose categorical. So I'll say SC, stem color. And then you can type the category. So green, I'll click the plus sign to create a new one. And then I'll type in the next category, purple. So now I have green and purple. So I'll save that. And then when I go back to my field and open that particular experiment, then now I have my stem color. So I can score it as either green, and then move to the next plot, it's purple, move to the next one, green. So that's the way we create new trait under Fieldbook app. Yeah. I hope I answered that question.
Yes, I hope to. So thank you very much, Dr. Dalid and Dr. Gaspar, and yeah, all the to you, Olufunke.
Yes. Thank you very much again, Dr. Dalid and Dr. Gasper. Moise is requesting that we turn on our cameras so that we can take a group picture. So please, if you can, please turn on your cameras and smile. And would also send the recording to all registrants. There's a comments or there's a link in the chat box for feedback. Please provide feedback. And after the pictures, I want it over to Ifeoluwa. So please turn on your cameras.
Okay.
Who's taking the pictures?
I have already
Fine.
taken it.
All right.
It's okay. You can go.
All right. Ifeoluwa, over to you.
Thank you very much, Olufunke. I hope we can all hear me. Just to be sure.
Yes, we can.
Thank you very much. We want to appreciate everyone for joining us in the fourth webinar. We want to say a big thank you to Dr. Dali, Dr. Gaspar for creating time to be part of this fourth webinar. And thank you for managing the time. We know for this kind of training, for most organizations, take 1 week to actually organize this kind of training and particularly be hands-on. We want to say thank you very much for delivering even at the shortest possible time, and you delivered so well. And we believe that for every participant that are part of this webinar, we are going to utilize all that has been taught. Thank you so much. We are very grateful. And to all our participants, we are also very grateful for joining. I could see some of our teachers from Nigeria and from other parts of Africa. And this shows our incredible turnout. Thank you very much for being part of this webinar. And we would also want to encourage that please let it not just end here. It is very, very important that we encourage the students that are under us, as well as the students that are part of the webinar too. Let's utilize the field book. The field book can be utilized even offline. You can get to make use of the field book. And it's like Dr. Dalid mentioned that for data integrity, as well as for you to have your data safe, it is important that you explore these electronic devices to be able to save your data on cloud in order for you to avoid stories that touches the heart. So it's very, very important. Yeah, so a very quick one. The next course of action is the symposium. I hope we're excited to hear about that. And so it's very important that we register for the symposium. The symposium is coming up on the 19th of September. That's 19th of next month. Very, very close. So we'll encourage us to please register for the symposium. It promises to be awesome. We're going to learn from so many presenters, so many research to explore, so many work that have been done throughout the globe. And this is a platform that encourages African students, boost up their confidence, encourage them to present what they do. You know, we always have this idea that we are being shadowboxed because of where we are coming from. However, we do quite a whole lot of quality research, but we don't get to present it out there. This is an opportunity for you to present what you do, even in this symposium. So for the symposium, the same link provided for the submission of abstracts is the same link you register. Provided on our screen, I hope you can see on the screen the barcode. You can just scan the barcode and you can register. The deadline for this abstract submission was on the 31st of August. Please, if you still have your abstract not submitted yet, we're pleading 31st of August is the deadline. Once you submit afterwards, it won't be honored. And for as many persons who will be selected to do the oral presentation, to also interest you to know that there are cash prizes. Yes, there are cash prizes, $100 for the first place in the graduates category, and $75 for second place in the graduate category, as well as $50 for third place in the graduate category. Also from the undergraduate category, we have the first place having $50, second place $35, and third place $25. The theme for this, the symposium, I know for many of us that have come across theme for the symposium, please, we are pleading, do not be scared, because we know so many persons have wondered, oh, I've not been involved in any smart-driven solutions as far as the symposium is concerned. So please, we know we're talking about precision agriculture for crop improvement. And for your submission, you might be looking at it that there is nothing close to precision agriculture for the research you do. However, it could have some innovations that are tilting towards that direction. Please do well to still submit your abstract, as it would also be considered to be looked on if it brings up something innovative, delving towards the precision agriculture. Is something that can be considered for the symposium. So please, we're encouraging us, let's do well to submit our abstract for this. And then finally, we would want to encourage us, please follow us on our LinkedIn handle. Just type in on your search button on LinkedIn, African Plant Science Symposium Series, and it will pop up. Share the link on the chat box. You could also get to click on the link so you can follow us on LinkedIn. And we will be dropping the link as well for the feedback. It's very important that we get feedback on every webinar series that is being done. It's to encourage us to do better and to do more. And we should also engage the comment sections on the LinkedIn post to be able to give feedback. Oh, this is what we should improve on as for the webinars. This is what we should improve on for the symposium. It goes a long way to help the organizing committee. Thank you very much. We really appreciate the turn up. God bless you, and we hope to see you at the symposium on 19th of September. Once again, Dr. Dalid, Dr. Gaspar, thank you so much for being part of this webinar series. Thank you for the time. We are grateful. We hope to have subsequent meetings with you where you facilitate other possible softwares that we could utilize in order to improve or enhance experiment on the field and even on the laboratory. Thank you so much, and God bless. So I hand over to Samuel. He probably has something to comment.
Actually, thank you so much, Ifeoluwa. There's nothing. I wish everybody a blessed day, and I hope to see, just like what Ifeoluwa said. Please do well to compete. Submit your abstracts to compete for the student competition. There's a huge prize there. Aside from the prizes, it also helps to boost your communication skills, talking to scientific audience also. Aside from that, see you all September 19, and do well also to register for the event, even if you are not competing. Thank you so much. Again, thank you to the speakers. Have a blessed day.
Thank you.